toxicity, excretion. These are very well known in pharmacokinetics, especially pharmacokinetics. Actually, what pharmacokinetics is all about. Yeah. And so, but right now it's kind of a sketch, so we are going to publish it as well. We are calling it NeuroAdmet. The first is NeuroClick, and the second is NeuroAdmet, and the third is NeuroAI, and some in the middle is NeuroChem. We have a lot of stuff here. Okay, so let's talk about the AI thing we are trying to develop, and this is the AI blood-brain barrier model. So right now we are trying to develop the model which is comprising the biggest database and also very precise in terms of the calculation of the blood, the log BB, the parameter which will give you the information how the substance will permeate through the blood-brain barrier model. There are different models so far in the literature, as you can see. People are publishing starting from 2018 till now. But most of them, I'm not sure whether they sometimes you have the publication, you would like to access it, you cannot. So it's kind of frustrating and unreliable. And most of the models, they're either not available to the public or they're commercial. Then you have to pay the money to see the code, or maybe just to do the trial. And here, the people are claiming that they have the model, they have the classification models, because you have in the AI, of course, you're doing as well, probably on a regular basis, either the classification or regression. Here we're also doing more like classification models. So we are clustering the chemical space of blood-brain barrier molecules, either the blood BBB plus or the BB minus, and see how they permeate. And then ROC curve, it's a standard procedure what we have. But so far, as I already mentioned, this is the drawback of all the models we have. So the main problem that the models are not really they are unavailable, or they are unreliable in terms of you cannot reproduce the results. How they are reliable if you cannot reproduce the result. Therefore, we try to come up with the open source model. It's already downloaded, can be downloaded from the internet. It's quite robust because we were using different algorithms, different open source algorithms, like boosting algorithms, random forest algorithm, as you can see here, and neural networks, and we were also trying different descriptors. Usually for chemoinformatics, the golden standard, we are trying to structure the chemical space. It's RDKit on a regular basis we are using it. And we have different descriptors, all of them are also available to the public. And we are using different variation of the descriptors to come up to the better optimized value for the precision here. As you can see, some models, they're quite successful than the others. So, as I already mentioned, this is published. You can use it for the boosting models. They are working the best right now. And the database is quite, I think this is right now this is the largest as well. Because we have kind of benefits. We have the laboratory. Professor Foster has a robotics facility where we can actually calculate the blood-brain barrier experimentally. So then we just integrating this particular information in a model. And on the other hand, in Germany they have a facility, high robotics facility in the University of Würzburg, and Fraunhofer Institute of Biomedical Research, where we also can utilize this high throughput robotic facility to probably to go beyond this particular number, maybe up to 100,000 molecules. But of course it's not easy, but we are trying to improve it. And so this is it. We are working on the database right now. implementing it, there will be the second version, which we hope will be more precise. And this is just a couple of words from my previous experiments where I was doing a lot on the viruses. This is the virology. I'm trying to combine with bioinformatics here molecular modeling, using some logistic curves. This is the logistic function. So actually, the result is quite frustrating, but the work was done for many years, but sometimes it happens in science. So what is the idea? The idea is that we have different viruses. They're called retroviruses, and in the retrovirus family you have lentiviruses, and the formiviruses, and the lentiviruses causing the HIV, and the formiviruses, they're not causing any pathology, and they're very widely used for the gene transfer between different cells. So you can transfect different cell lines if you want, and even you can use for the gene therapy, so they are not really pathogenic. And Professor Redfeld in Germany is the main person who is doing this research for many years. Like for 30 years, he's the one who developed actually the in vitro assays for particular formiviral vectors. And these are vectors coming from his lab. I was just trying to optimize this vector, and the idea is if you look at the lentiviral vectors, because lentiviral vectors are well known, then you can easily transfect dividing and non-dividing cells, because they can go to the cell, then the RNA will be converted to the cDNA, and there is a pro-integration complex will go to the nuclear core, because you don't need a breakup of the nucleus, and the virus will be integrated in the genome because it has the integrase protein, which is not the case for the formivirus. And at the time we thought if we will substitute some genetic elements, we might come up to the same mechanism. And in a lentivirus you have flat, in a formivirus you have gap, but you have some particular things. They're defined. You have so-called polypurine tract in the formiviruses and polypurine tract in the lentiviruses, and because of terminal sequence, central terminal sequence, it will create a flat, which is very important to form the pre-integration complex. And once the pre-integration complex is formed, it will go to the nucleus, through the pore. And I was just doing a lot of substitution to see how is it really possible to create a vector which is very efficient to infect non-divided cells like neurons. And that's what I did. And this is the final experiments. We have the reporter here, the EGFP, and as you can see, if you have an antiviral vector, it's very efficient, almost 100%. And when you get down the road, you will impairing the transfection efficiency of the cells to express the GFP, which means you cannot really substitute without the severe damaging the replication machinery of the virus, despite the fact that maybe these particular elements are promising if we will substitute different gene therapy vectors. Yeah, but this was the result, and so far the project was still on hold. Yeah. Okay, this is also kind of interesting thing, and